about the X4 towards the end, and these are for small cell counting. So today we'll go over some of the software screens, some of the features, some of the applications, and then I actually have a yeast sample that we're going to count on the system here. So my email is there below also. If you have any questions after the webinar, feel free to send me an email. So first we'll go over a brief introduction to the X2. What is the X2? We'll get familiar with the software. We will perform an AOPI viability assay using Yeast. And then we'll talk about some additionally supported assays that are available on the X1, X2, and X4. So the X2 technology, it has fixed dual fluorescent channels. You see there are 535 and 660. Those are the two filters that are inside the unit, and they are not exchangeable. Okay, for the X1, we have a single filter, okay, and that's a 660 filter only. Both the X1, X2, and X4 are suitable for yeast concentration and viability measurements, and those can be achieved a couple different ways, and I have a slide at the end that we'll review. The recommended viability measurements are performed using AOPI, which is a nuclear dye. As you all know, yeast have a nucleus, and this can be performed using the Cellometer ViaStain kit for yeast. It's a very simple process, and again, we'll cover that in just a minute. Other assays that are available on the X2 are a GFP transfection assay. I've worked with a few users in the past who have yeast transfected with GFP. And you see there in parentheses, strong signal GFP. The X2 is not designed for detecting weak GFP signals. Going along the same lines, we can do a GFP PI, which gives you viability, so you can get the percentage of GFP positive cells as well as the viability by staining with PI. And again, strong signal GFP is required. And lastly, yeast cell cycle can be performed since the X2 has FCS Express software that can be used with the system to generate the cell cycle histograms. So like I said, the X2 has two fluorescent optics modules. Those are fixed. And you can see there the excitation and the emission for the two filter modules. And the X1 is not listed here, but the X1, if you look at the second column, the 540 nanometer excitation 660 emission, that is the same configuration for the X1 filter module. So that takes us through our introduction to the X2, and now we'll go over the X2 software. So you can see there, there's a little GoToMeeting thing in there. I apologize for that. Circled in red, this is our assay selection area, and I'll show you in actual software. But this is where you would choose your assay. So if you're performing a GFP or an AOPI assay, you would select that from a drop-down menu there. We have our sample ID, which is where you can set a unique sample ID. So if you're in brewing and you would like to set a sample ID based on the batch or what fermenting tank you're taking the samples from, you can do that there. You can also log a user ID so that you know if you're performing QC, for example, you can log whomever performed the QC for that day or that run. Here's our preview image area where you would preview your bright field and fluorescence. I'm sorry, the formatting's a little off here. And then on the right is our image navigation, so we can view images. We can go through the 4 images that are taken. We can zoom in, and we can navigate bright field and fluorescence. And then, sorry again about the formatting, up at the top is where we can go over our data management features, and I'll show that within the software. So here's our results screen. Okay, this is what will appear after you perform the count, and you can see here our assay. We've selected yeast AOPI viability. The 2 cell types that the software have linked to the assay are shown there. We have our date and time stamp. We have our sample ID, so that's where you can log in, like I said, the fermenting tank or the whatever batch or sample you'll be processing. And a dilution factor, which reflects a 4-fold dilution, which is what's standard for our yeast viability kit. And then we have our results. So you have a count, which gives you a total number of yeast cells counted, as well as a live and dead number. concentrations for those populations, and also an average diameter. And then last but not least, we have our viability percentage reported, which just takes the live and the dead and calculates the viability based on those two populations. So now we'll go over the AOPI viability assay. Okay, there we go. So quickly, we're going to review the AOPI video and how this works. So this can also be found on our website. Let's get this playing for you. Again, technical difficulties. Let me go to our website. I'll bring that up there. So our website has a lot of information on it, and one thing we have are a lot of videos. So here's a video of how the mechanism of AOPI works.
AOPI assay for cell concentration and viability. This assay is most suitable for primary cell samples, such as white blood cells, splenocytes, and lymphocytes, which contain debris and unwanted non-nucleated cell types, such as red blood cells. Acridine orange and propydium iodide are nucleic acid binding dyes. They produce strong fluorescence after binding to DNA. AO is permeable to both live and dead cells. PI is only permeable to dead cells. Cells are mixed with both acridine orange and propydium iodide. Channel 1 of the cellometer is configured with an optical module with blue excitation and green emission. For a live cell, AO enters the cell membrane and binds to its DNA. Under blue excitation, strong green fluorescence is emitted and detected. PI is excluded from a live cell. Both AO and PI enter a dead cell through the damaged cell membrane and bind to its DNA. Under blue excitation, the green fluorescence by AO is absorbed by PI, producing an extremely weak green signal. Channel 2 of the cellometer has green excitation to detect red emission. Live cells exclude PI. Without its DNA binding, there is no detectable red light from live cells. For a dead cell, PI enters the damaged cell membrane and binds to its DNA, producing a very strong red fluorescence. To perform a cellometer AOPI assay, load stained cells into a counting chamber. Insert the chamber into cellometer. Cellometer automatically performs cell image acquisition and imaging analysis. Typical assay time is less than 60 seconds. Cellometer can also perform many other dual fluorescence viability assays. Explore our website at Nexcelom.com for more information.
All right, so thank you for watching that. So it mentioned splenocytes, it mentioned red blood cells. Those aren't the types of cell types we would be working with for the X2. We would be working with yeast or even platelets or polystyrene beads that are very small. And for yeast in particular, many samples I've worked with, depending on what stage they are in their fermentation, or if you're working with biofield samples, can tend to have other things in there, debris, just junk in the sample. So even though it's not red blood cells per se, it's still debris. And by using AOPI, it will exclude that debris and give you much more accurate measurements of concentration and viability. So the AOPI protocol for yeast using our ViaStain kit So we have a what's called a dilution buffer. Okay, and this is what facilitates the acridine orange, which stains your live cells. It facilitates binding of that to your live cells. So we first take our yeast sample. We mix at a 1-to-1 ratio, so 20 microliters with 20 microliters of the yeast dilution buffer. Next, we mix the diluted yeast sample with the AOPI staining solution, which is optimized for yeast cells in that kit. And we mix that, again, at a 1-to-1 ratio. So we can take 20 microliters of a diluted yeast, mix to 20 microliters of the AOPI solution. We pipette 20 microliters of that onto our disposable counting chamber, insert the slide, select our assay from the drop-down menu, and count and acquire our results. So now I'm going to quickly demonstrate the viability assay using the Cellometer X2. So let me see. All right. So here is our software. Okay, you can see Cellometer X2. Over here we have our assay drop-down menu. Okay, and for this sample, I'm going to choose our yeast AOPI viability. All right. So the next thing we will do is we'll set our sample ID and user ID. So I'm just going to write Ben P, and I'm going to give this a yeast viability number 1. We'll save that. You can see that change is reflected down at the bottom. And the next thing we're going to do is preview B1. You can hear the instrument switching filters. All right. So right now the image is out of focus. So as I adjust the focus, we will find the proper image. Okay, come back a little bit, right about there. Okay, and then I'm going to move over to the right side of our image. I'm just going to zoom out. Okay. Actually, there's some better looking cells over here. All right. So what we're going to do now is we're going to take a look at our F1. All right. These are our live cells. And then we'll look at our F2. And these will be our dead cells. And you're going to hear the filters switch again. All right, this is a fresh sample, so we're probably not going to see any dead cells. But you can see down here there's exposure times, 700 milliseconds for F1, which is our live channel, 6,000, which is 6 seconds, for our F2, which is our dead channel. Those can be optimized by clicking this little pencil up here. Okay, so and you can see here fluorescent exposure for our F1 image, and then fluorescent exposure for our F2 image. Any of these parameters that you see, I can assist you with optimizing. So we'll save this. Okay, we'll go back and we'll preview our bright field again. All right, and you can see now, if we zoom out, we can go ahead and we'll count our images. So the software and the instrument will now acquire the images, 4 images. Okay, and it's going to switch back between bright field 1 and fluorescent 1, and then it will switch over to bright field 2 and fluorescent 2, and do that 4 times. You can see at the bottom, it tells you the status. So it's taking fluorescent 2 image C. So we know that it's nearly complete. Now we'll count. You can see the counting goes very quickly. All right, and here's our results screen. This is what we saw earlier. Okay, so we have our total, very dilute sample. Our dilution factor is 4, reflecting the dilutions done to the sample. Okay, so our concentration, no dead cells, which is what we saw, So the concentration is about 1 million cells per mil. Average size is about 4.5 microns. Viability of 100%, which is good. So here are the images we acquired. We can actually go back and we can review what's counted. Okay, so you can see that it will circle the cells that it counts. All right. Up here at the top of the page, we can look at a size histogram. No diameter data. We'll look at, that's a new one. Anyways, we can look at a size histogram. Okay, we can also use a sample adjustment calculator. Okay, and this is useful to adjust any samples if you're going to grow the yeast and you want to determine a target concentration or number of cells. This will tell you how to adjust the sample based on the results you obtained. Okay, but the most useful part of using an image-based system is being able to review what's counted and verify that what is counted is correct, and that all the fluorescent signal in this case is circled. So let's go back to our presentation. All right, so the final one is the additional assays that are supported on the X2, and here this is a list of the complete assays that are supported on the Cellometer X2. If we want to talk about the X1, what's supported here is the yeast viability by Brightfield, so this top assay here. Okay. Also, what's supported is the yeast cell cycle analysis, since we'd be using propydium iodide, and the filter configuration for the X1 fits that configuration. So cell cycle analysis and viability by Brightfield and propydium iodide are the two assays here that are supported on the X1. On the X2, you can see there's many, many more. Yeast vitality, a couple different ways to detect viability, and also, as you mentioned earlier, the GFP transfection assay. Other assays that can be performed on the X2, since the X2 is designed for small cell counting, platelet concentration by Brightfield only. You can also perform platelet concentration by fluorescence using the calcium AM kit. We have a calcium AM kit that we have here that works great for platelets. And then other small particles, and you see listed there, polystyrene beads. I worked with some groups before counting some small polystyrene beads that, again, are below 5 micron and can be counted on the X2. So lastly, we have some references here. And this, I mentioned in the beginning, I talk about X4. The X1 and X2 systems are very new to Nexcelom. They're about 6 months since they've been released. So currently, all of our references regarding small cells are for the X4, which is our older small particle counter. So X4 has user-exchangeable filter modules, and there's 7 publications here that can be found on our website that reference the Cellometer X4. So with that said, I'd like to open it up for questions. So go ahead and ask whatever questions you might have.
Great. Thank you, Ben. So as I had mentioned before, everybody is muted. So if you do have any questions, please feel free to type them into the questions section on your dashboard on your webinar and GoToWebinar screen. We'll be happy to answer those. Questions may just request clarification. Maybe you have a specific type of small cell that you want more information about to see which instrument would be best or which kind of assay you would be interested in. We'll be happy to answer those right now. So we do have one question, and it's saying, what other instruments are optimized for small cells for counting and viability and things of that nature? So we talked specifically about the X1, X2 today. Ben briefly mentioned the X4. So the question is, what other instruments might also be useful for small cells?
Yeah, great question. Thanks, Sarah. So the instruments that are optimized for small cell counting are the Cellometer M10, M as in Mary. That is a Brightfield only unit. Okay, so there's no fluorescence no fluorescent assays that unit can perform. There is the Cellometer X4, as we mentioned, that has user exchangeable filter modules. It's a single channel, single fluorescent channel instrument. And then we have the X1 and the X2. All other cellometers are designed for counting larger cells, mammalian cells, that have a size of approximately 6 micron all the way up to like adipocytes, which can be 250 microns in diameter.
Great. We have another question, and it says, one of the yeasts that we use is quite dim with the AO stain. Would you recommend increasing the exposure time?
Yeah, that's a great question, and I have seen that in the past. Some yeast samples require a little bit of a different protocol than just simply diluting and instantly running the sample. What I have found to be successful with those types of samples is actually incubating the AOPI at room temperature with the sample. So you would perform the same steps, take your yeast, dilute with the yeast dilution buffer, and then take 20 microliters and dilute with the AOPI, and let that sit. I've seen it work well at 3 minutes. I've seen it work well at 5 minutes, at 7 minutes. So it might just be that the AO has not had enough time to bind to the DNA inside the yeast cells. So trying just a time course study and seeing how that affects the signal would be a good start. And then you can always contact me. If you have internet access, we can do a remote desktop session. But if that does not work, we can support you to get it to look better for you. And increasing the exposure time may help, but generally an exposure time of 1,000 milliseconds or even 1,100 is kind of the maximum exposure that you want to be at for the AOPI assay. And again, that's a maximum of about 1,100 milliseconds for the F1 channel. Generally, it should be around 700 to 900.
Great. Tom, I just sort of summarized what Ben just said, and I sent it to you privately, and it also includes Ben's email right there. So as he mentioned, please feel free to reach out to him if you want to do a session or a WebEx to help troubleshoot and work through it with him. Do we have any other questions?
Mm-hmm.
Well, if there are no other questions, we can go ahead and conclude this session for today. Again, I want to thank everybody for joining us today. Next month, our topics are AOPI versus Trypan Blue, and also our bright field instruments focusing on the Auto 1000. Oh, it does look like we have another question. Oh, you're welcome, Franklin. It wasn't really a question. So we will be sending out invitations for the next webinar next month. As always, it's the 4th Friday of every month at 11 o'clock. Again, this has been recorded, so you can always view the recording at the same link you used to register. And if you do have any questions that come up after the fact, please feel free to reach out to Ben at bparadis@nexcelom.com, or you can always email support@nexcelom.com. Thank you, everybody, and have a great day.